Hey guys, it's been a long time since we had a video, so why not throw one at you right after your quiz? Hopefully the quiz went well. Couple of things I want to talk about. Today, we are talking about meiosis. So we were only talking about the cells that are involved with making you. So these are known as sex cells. And there are a few things that I want to make sure I go over in this introduction before I even progress into this brand new information. Previously, we have talked about mitosis. Remember, mitosis is the process of replicating a cell and creating an identical cell. And your body, like most of your body, like basically like 95% of your body is made up of these cells known as diploid cells. And these undergo the processes for your liver, for the functions of you breathing and everything else. Now we have these special cells known as sex cells, and these are haploid cells, meaning they only contain half of the genetic information. So if you are a male, you have sex cells known as sperm. And if you are a female, you have sex cells, and these are known as your eggs. And they have half the genetic information because when those sperm and that egg meets each other, they're going to create a diploid cell, and that's what divides and helps create a baby or a zygote. And we'll get into all of those specific names later. The one thing I did want to make sure I covered at the very beginning of this is a chromosome and its structure, because you may not remember. So remember, we have 46 total chromosomes, and those 46 chromosomes pair up into 23 pairs. In mitosis, those chromosomes will replicate, in interphase, they replicate each other. So we now have 92 chromosomes instead of 46 because of the fact that we're making two diploid cells, meaning that those two cells are both going to contain 46 chromosomes. So I'm going to draw a chromosome for you really quick. So here is one chromosome hanging out, doing its thing, one little chromosome. And one comes from your mom, one comes from your dad. But remember, during interphase, during DNA replication, this chromosome, we also have called it a chromatid. Chromosome and chromatid can be used interchangeably. That chromosome will replicate itself, and it'll create a genetically identical copy of itself, and they come together, and this is also what's known as a chromosome. That chromosome will then be attached in the middle by a centromere. Remember, these chromatids are genetically identical to one another. Sister chromatids, genetically identical to one another. That's really important because now we're going to start talking about homologous chromosomes. And what that means is that they are essentially the same chromosome in the fact that they have the same kind of DNA, but one is from your mom and one is from your dad. We have not focused on what that looks like when they come together and separate and what creates the individual person. And so I want to make sure you have all of those details fine-tuned. And if you still don't, we're going to talk about this again in class. Oh, I don't have an objective slide. That's okay. We'll talk about what you should know by the end of this video in class. Remember, this is part 1 of the video, so there is some stuff in the next video that will relate all of this information. Really, in this video, we're only focusing on meiosis and that process. All right, first thing, the process by which organisms give rise to offspring is known as reproduction. So whenever an organism reproduces, it's creating an offspring. And it's a great thing that organisms reproduce. The reason for that is because the species would not be able to continue or live if you can reproduce. Now, there are two types of reproduction. One is known as asexual. The other is known as sexual. Oh, I didn't capitalize the sexual, and that's annoying. But anyway, asexual reproduction is generally done by prokaryotes and very single-celled organisms in comparison to sexual reproduction, which is done by eukaryotes. So let's spend a little bit of time talking about asexual reproductive. This only involves one parent, so a single parent, and results in the offspring that is genetically identical to each other and to the parent. So a lot of genetic diversity is not offered here because of the fact that you have one single parent and it is genetically cloning itself almost. So humans do not do this. This is more, again, of the process of like some eukaryotic cells, but mostly it's just prokaryotic cells, so bacteria. Binary fission is one type, fragmentation is another type, and then the third type is known as budding. So the first one we're going to talk about, I'm going to put a 1 there instead of a dot. Binary fission is where a parent cell splits into two identical cells of the same size. So basically, we talked about the binary fission in the last video. It's basically just taking itself and it's splitting. I mean, there's really nothing else to it. Fragmentation, this is when the parent organism, so one parent again, breaks into fragments or pieces, and each fragment develops into its new organism. Good example of doing this is a starfish. So if you were to cut the leg off of a starfish, that one leg will eventually develop into a brand new starfish. So it's kind of cool, very, very evolutionary favored. And keep in mind that starfish is a eukaryote, so not all types of asexual reproduction are strictly just for prokaryotes. Mostly, but not all. And then the final type of, I didn't put a 2 there. And then the final type of asexual reproduction is known as budding. And this occurs when that one parent cell, it forms almost like a little bubble-like thing. It's like a little buddy. And that little bubble-like thing will stay with the parent cell, but it just grows and develops. Eventually, that little bud becomes fully developed, and then it just breaks away and it says, okay, bye, Mom. I'm leaving. I'm going to go do my own thing. This is very, very common among yeast. So I know it's weird to think that yeast is actually a living thing, but yes, it most definitely is. Great advantages to asexual reproduction is the fact that it happens super quick. For many organisms, this is a great advantage. For example, bacteria. You can have, and the reason why you get sick so quickly if somebody were to cough on you is the fact that bacteria can divide so many times per hour. So under ideal conditions, 100 bacteria can divide and to produce millions of bacterial cells in just a few hours. However, not all bacteria lives in this perfect ideal conditions. That's why it creates so much of it, because the chances of survival are very slim or very small. So it does this rapid division with knowing full well that most of that offspring is not going to survive. If bacteria divided that quickly and survived, the world would be plagued in bacteria and there would be very little life, if any. So it's a very good thing that the bacteria doesn't actually live in ideal conditions all the time. So there are a lot of checkpoints to keep the reproduction of bacteria and those very small organisms in check. The next type of reproduction is known as sexual reproduction, and this involves two partners. So in sexual reproduction, parents produce reproductive cells, and these are called gametes. So your sex cells is what I've been calling them so far. They are known as gametes, and when they unite, they form an offspring. Gametes, it's really important. So those sex cells, they only contain half of the number of chromosomes found in other cells of the organism. So basically all of your body, they have 46 chromosomes. Your sex cells, or your gametes, only hold 23. And these are known as haploid. The way that gametes are formed is by a process known as meiosis. So it's similar to mitosis in the fact that they have the same stages, but meiosis is just a little bit different because we are now talking about genetic diversity. You don't look 100% like your brother. You don't look 100% like your mom or dad because of the fact that you have genetic diversity. And that's a huge thing in meiosis. And we're going to talk about that here in a second. So the process in which two gametes unite is called fertilization. When fertilization is successful, you create what's known as a zygote. A zygote is a diploid cell, which means that it has twice the number of chromosomes as a gamete. So I always have been calling the zygote a baby, but the very first union of the sperm and the egg and a successful fertilization, that is called a zygote. Eventually, that zygote forms and multiplies and it becomes a baby, but there are a few names before that process occurs. So again, remember, we are now not focusing on diploid cells. So we are not going from 46 to 9, so from 46 cells in interphase, and then replication 92 in that one cell divides and becomes two genetically identical cells. That is mitosis, and that creates diploid cells. In meiosis, we are focusing on taking that 46 and undergoing two processes of cell division where we create 4 haploid cells. So for every one cell, we are creating 4 in the process, and that's meiosis, and these have 23 chromosomes each. And the fun part about that is the fact that the chromosomes that you get, you don't know if you're making chromosomes with mostly just moms or dads. genetics. So that's why you can get kids that look like mine. So remember, Jace looks exactly like me. He's darker complected. He has dark hair. He is essentially my twin. Whereas Jackson, on the other hand, looks nothing like me. Very light complected, blonde hair. His eyes are hazel, so he looks a lot more like Barry and has those features. So that's where that genetic diversity comes from. It's a great thing because, I mean, it really goes to show with just my family alone. And I don't want you to think that genetic diversity is just strictly, oh, mom and dad, because there's a lot of other factors that go into it compared to just dominant and recessive. But it really, meiosis is the big key factor on why humans are genetically diverse. This also leads into the idea that some, like in a family, you'll have one brother that's completely healthy. I mean, everything is fine. And then the parents could have another son, and that son maybe comes out with some kind of genetic defect that causes that son to not be as healthy. And so that's where meiosis comes to play. Big things right now is you need to know is that meiosis is done in two processes known as meiosis 1 and not meiosis 2. All of the phases or stages, remember I use those interchangeably, names are the exact same as mitosis, but things that happen in those phases and stages is a little bit different. Again, we are going from 46 chromosomes to 23, so we are reducing the number of chromosomes in half. During meiosis, homologous chromosomes separate, haploid cells form and have only one chromosome from each pair. All right, remember from the introduction when I talked about a chromosome, remember a chromosome is like that one little leg. We have been calling it a chromatid, but you can interchangeably hear it called a chromosome, okay? And when it replicates or becomes that X that we know so well, so remember here's the centromere, it replicates and becomes that X. These two are sister chromatids. They are genetically identical to one another. Okay? Please make sure you understand that. These are genetically identical. So this does not mean that one is from your mom, one is from your dad. This is just like one chromosome from your mom and it has doubled or it's replicated. So now these are two chromosomes from your mom. Okay? So in interphase, before the process of splitting the nucleus even happens in meiosis, in interphase, we have homologous chromosomes. Okay, so one from your mom, one from your dad. Homologous chromosomes means that they're the same type, but they are not genetically identical to one another. Sister chromatids, these guys here, are genetically identical to one another, but homologous chromosomes are not. They just have the same information on the same chromosome. Okay, so before meiosis 1 begins after DNA replicates during interphase. So that means if we started with 2 chromosomes that are homologous to one another, they're going to create 4 chromosomes, if you will. And I'll show you a picture of what I mean by this in class because it doesn't really sound like it makes sense. But remember, homologous chromosomes, one from your mom, one from your dad. Mom will replicate to create the X structure that you know so well, and then dad will replicate and create that X structure that's bound by that centromere, and then we have now 4 chromosomes or 2 of the X's. Okay, so the chromatid and the chromosomes, you'll hear those words interchangeable. So again, my DNA replication has happened. We have homologous chromosomes that are together. In prophase 1, the nuclear envelope breaks down, and that's what's happening in prophase 1 in mitosis. The chromosomes condense and they form into those X structures. Centrioles start to move to the opposite sides of the cell. Spindle forms. The biggest and most important thing that happens here is that homologous chromosomes pair up. This is very unique to prophase 1. Okay, so homologous chromosomes, not chromatids. Homologous chromosomes, they pair up. Okay, and they're like, hey, you look like me. You and I are going to be friends. Okay, so that's basically what it is. They're alike each other. And so, you know, like attracts like. So things that are similar to one another, they're going to come together and they're going to say, yep, we belong together and we're going to continue this journey. And again, that is very unique to prophase 1. So in prophase of mitosis and meiosis 2, homologous chromosomes do not form pairs in this way. So in only prophase 1 of meiosis 1 do homologous chromosomes pair up. A great reason why they pair up is the fact that crossing over begins. And what this means is that pieces of genetic information from one of the chromosomes could pass on to another chromosome. And I'll show you that in class as well. So now the rest of the stages kind of go along with everything. Remember, we are in homologous chromosome pairs. So those homologous chromosomes, they line up in the middle. So the spindle fibers attach, they line up in the middle. Anaphase 1, the spindle fibers shorten and the chromosomes of each homologous pair separate to each side. So right now, everything is in those X's because remember, we duplicated our DNA. Each X goes to one side of the cells. Telophase happens, and what happens is the spindle fiber breaks down, nuclear membrane reforms itself, but you have chromosomes on each side. You have now essentially created two daughter cells, each with chromosomes. Now, it's important to know that once cytokinesis happens, we have formed two haploid daughter cells, each with 46 chromosomes. Now, that's not what we need when it comes to meiosis. We are trying to create a haploid cell. So that is why meiosis 2 has to happen. Because what we've basically done in meiosis 1 is the homologous chromosomes have gotten together. They have randomly assorted the chromosomes in the fact that you get genetic diversity and crossing over. But the daughter cells that we've created each have a random assortment of chromosomes, one from each homologous pair. Okay, and I'm going to show you pictures because I can already feel your brains like going crazy right now because I'm not drawing this and a lot of us are visual learners. All right, but that's meiosis 1. We've created two haploid daughter cells. They have enough chromosomes right now, but we know that we need to get to haploid cells in meiosis. So here we have meiosis 2. It's basically the splitting, but now we're creating 4 instead of 2. All right, so in prophase 2, the nuclear envelope breaks down yet again. So if you don't remember, recap from meiosis 1. We have created two haploid daughter cells that have all this genetic diversity. We don't know it's just a random assortment of chromosomes, one from mom, one from dad. So that means that one cell could essentially have everything given to them just randomly from mom. And one cell could have everything given to them randomly from mom and dad. So those two cells, again, we're taking those, we're breaking down the envelope, spindle fiber forms again, and each haploid daughter cell from meiosis 1 begins to undergo that same process. And metaphase 2, spindle fibers line up, sister chromatids of each chromosome line up in the middle. You have that middle lining up again. In anaphase, the sister chromatids separate, moving to opposite ends of the poles. Telophase 2, we then form 4 new nuclear membranes. And the cytoplasm of each cell divides in cytokinesis, creating 4 haploid cells. I am going to provide a picture to you because, again, I can already see, like, some of you're just freaking out right now. That's okay. This is not an easy process. We spent so much time on mitosis, learning that, and now we're basically putting mitosis on steroids right now. So again, we will talk about this in much more detail in class. I'm not super worried that you understand or memorize this right now. The biggest thing is that you need to know is that by the end of meiosis 2, the sex cells, or the cell that was the initial cell, has now formed into 4 haploid daughter cells, each with a chromosome that has new genetic material that's very special to itself. So the chances of you creating identical sex cells is like almost 0%. Very, very small. And so you may be thinking, well, what about identical twins? Well, identical twins, what they actually are is one egg that just randomly split. One egg one egg fertilized with one sperm, and it just randomly split. And you have identical twins. My brother and I happen to be twins, so we are 2 different eggs and 2 different sperm that came together. So essentially, we're just brother and sister that happen to be born at the same time. We have nothing about our genetics that is identical in any sense of the way. We just happen to be born on the same exact day. All right. Again, if you are super confused, that's okay, because we are going to go through this in detail in class, and there is a part 2 to this video. So right now, you should just know that we have 4 haploid cells that are perfectly unique, and then we will learn about what happens to those cells after fertilization has occurred. If you have any questions, please ask me on Edpuzzle. Thanks.